today. I tried to get rid of as many movies as I could. Your patience with my technology. It's coming. Okay, so I was going to give some career advice. Well, because I've been thinking about this for a while. And there's two things that students never know. And I think I'm going to just be this nice person and tell you about it. So one thing is when you're emailing someone to work in their lab or to get research experience or anything where you're asking them for something, have someone look over your email first just to make sure it's written appropriately, because there are certain things you don't want to do. Like for one, you want to be formal, dear Dr. So-and-so, not hey, none of that. You guys know that. You guys know. But the one thing that I don't think people realize, because I've had really great students see this, is don't tell the person you're asking for something that you want the thing from them, like, hi, I want to work or shadow with you or whatever, because I have to. I need these credits. Help me. Because no one wants that. No one wants to help someone who doesn't want to be there, right? So just think about it when you're writing these emails to someone. Just say you always want to be, and I've gotten great emails too of like, I love your research. No, they don't have to say that. But like, I know your research. I want to work with this kind of stuff. So just kind of keep in mind as you guys are getting ready to go off into the world and you're going to be asking people for lots of things. Flattery. Flattery, don't make it sound like that person is, something you have to deal with. Okay, let's see if we can make this happen. Okay, so exams will be back by the end of the week. I'm done with most of them. I just made a faux pas and forgot to bring my computer home last night. So definitely by the end of the week, things are looking good. People are doing really well. So, and people who've really been struggling have been doing well. So that makes me happy. That's that. So let's just take 5 minutes to write. Okay, so microtubule stability is dependent on the GTP cap. Discuss. So talk about the GTP cap, the role it plays in microtubule stability. And so then consider this one situation. So you're doing an experiment, right? You add some tubulin dimer mutants that cannot hydrolyze GTP to your experiment. Predict the outcome in terms of microtubule stability and life. And so go ahead and do that for a few minutes. I know some people weren't here much last week. Hopefully you caught up on the lectures. Okay, so Gary, tell us what you were thinking of when I told you to discuss the GTP cap and microtubule stability.
I was not sure about the first part. I thought from the second part more that the hydrolyzed GTP would cause it to, I guess, to grow the microtubules, to make them bigger. So if you mutate that it cannot hydrolyze them, they won't grow, they'll just be collapsing.
Okay, so you're a little bit reversed. The GTP actually makes for strong binding. So if you can hydrolyze GTP, you would have the opposite, which would be strongly bound. But thank you for your input. Who else? So who wants to talk about the GTP cap and microtubule? Yep.
So like, doesn't it allows for like microtubule growth? Don't have GTP now, and then you'll have depolarization, and then doesn't it mutate at 2 feet?
Yeah. Yeah, exactly. So GTP cap means the subunits are GTP bound, rather than GDP bound. It promotes growth. When you don't have the GTP cap, then you have disassembly and eventually catastrophe. Yep. So if you add tubulin dimer mutants that cannot hydrolyze GTP, I just said it, but does anyone want to let me know what they were thinking with this second part of the question? Yes?
When you hydrolyze GTP, is that helping promote growth? It's helping it shrink.
Yep. So the question was, when you hydrolyze GTP, is that promoting growth? And so I said no. It's so think about these dimers. So remember, we've had a lot of nucleotide-bound proteins in this class, right? And what that nucleotide binding changes the overall shape of the protein, right? You got that concept. And so with the GTP-bound tubulin dimers, or ATP if you're thinking about actin, you're going to have strong binding. And then you'll have GTP hydrolysis just over time, and that just occurs after you have addition of subunit to the growing polymer. Over time, you'll get GTP hydrolysis. That will change the shape of your subunits, and that will into a conformation that binds less tightly to the polymer. And so that's why the D-bound subunits are more likely to leave rather than stay bound. So in this case, we should have what? Wherever the GTP is, that part will be growing? Yeah, because you can't hydrolyze, so GDP is going to fall apart. Here you can't hydrolyze, you're stuck in T-bound subunits. And so you're going to be growing and you're going to be stable. Yeah?
So, what if there's like, so you added GTP that can't be hydrolyzed, but there's GDP, wherever the GDP is happening, and there's GDP moving out, it's like a second, like, this is your chain, like one part is just, I won't say moving from GTP, but then the GDP over down to that point, and then.
So I think the question, you're having some confusion over if there's some already that's GDP bound. Okay, but that's not in the question. So you're overthinking it, but good scientists should always overthink things. So you guys are all great scientists. You all overthink things. So the question is, will you have shrinkage because of that GDP bound tubulin? That's your question. Okay, does anyone want to answer that? All these hands were up and they just went down. Well, so if you think about it, this GTP cap is what stabilizes microtubule, or makes it more likely to be growing. So the fact that all new subunits that are added are going to be irreversibly GTP bound, you might lose some of your GDP bound from your minus N, but you're going to have this, all the GTP bound subunits that are added will be stable because they'll never hydrolyze. Okay, yes?
Don't you have a critical concentration? So wouldn't you run out of GTP tubulin at some point?
So you wouldn't have constant nodes, but.
Right, so you're saying, okay, where do I fit in that concept of critical concentration with this question, right? Because once you're below your critical concentration, you should have disassembly, right? Well, the fact that you can't hydrolyze this GTP artificially is going to make it stable. Normally, you would have that hydrolysis, which is going to weaken the binding, and then it will start to disassociate over time. Yep, anyone? Yep. Would this be a bad thing? Oh, I like that question. Okay, so if we had GTP that could not be hydrolyzed in our cells, and it was limited to interaction only with tubulin. Obviously, GTP that can't be hydrolyzed should have many effects on the cell, right? But just talking about cytoskeletal proteins, is this a bad thing? Yes? Wouldn't that mean you couldn't dissociate microtubules, break down microtubules during mitosis? Right. Absolutely. So you'd die, actually. It would be really bad. People think stable is good, but when you have no stability, I mean, when you have everything in moderation. When you think about what microtubules do and how that dynamic instability is so important for all these different processes in cell biology, as soon as you get rid of that dynamic instability, you're going to have a cell that can't divide, can't respond to the environment. So, very good. Yep.
So is this like you're keeping at their size and associated with just an abundance of microtubules?
Basically, what you're doing is you're keeping microtubules the way they are.
Like if you like add so many microtubules up there, would it cause like apoptosis?
So if you had too many microtubules, would it cause apoptosis? It's not the microtubules itself that would, but its effects on cell cycle. You would trigger checkpoints in cell cycle because you wouldn't be able to segregate chromosomes properly. And so then you would have cell cycle arrest and probably apoptosis. Yeah. And so we can think about this as we move to the next one for your cell cycle in a few weeks. Good, lots of good questions. Okay, that was more than 5 minutes. We're just not going to think about it. Just everyone think positive things about my computer today. Here we go. I don't know what to do, guys. Get a new computer. This is like a new thing for this semester.
Space, why don't you just get one?
Why don't I get it? What? I have a whole. Yeah. I think it's more than that. I think something else is going on. Okay. Huh? It's dying. It's tired. My computer's tired. Okay. So you guys are awesome for putting up with this drama, but I really think it's important to record the lectures. I have a feeling if I didn't record lectures, we wouldn't have this. But you probably like the recordings more than putting up with this. So as I work through this, just be happy you have recordings. Okay, so far what we've done, key players, actin microtubules, intermediate filaments, accessory proteins. Major concepts in cytoskeletal dynamics, dynamic instability, filaments, producer subunits, nucleation, critical concentration, NTP, NDP, and its relation to that's ATP or GTP, and its relationship to subunit assembly and stability, and treadmilling. So these are major concepts you're going to start understanding. Yeah.
Since you were kind of on the topic of the recording, why is it like when you're on YouTube and you're watching videos, your recording's fine, but like your videos like shit?
Yeah, something funny. Oh, yeah, I don't even know what happened there because I noticed that too. And is that just recently?
That's like
Oh God. I think it has to do with something with how PowerPoint's running on this side. But email me and I'll call a professional. I'll call Rooter or someone. Okay. So this is just the concept of critical concentration. I'm starting where we started to leave off on Thursday because a lot of people weren't here. Critical concentration is confusing for people, And so really it just has to do with the fact that on either side of your polymer, you've got your plus end and your minus end. Each side has a critical concentration. And when the concentration of subunits in solution is higher than this concentration, then you're going to have assembly. And when it's lower, you're going to have disassembly. Okay? And this is all based on what's happening in a test tube when you have no accessory proteins. When you have accessory proteins, they manipulate all of this, and that's how we can control our cytoskeleton and our cells in response to the environment that uses the cell and all that. Critical concentration is lower for the plus end and higher for the minus end. Hopefully everyone will have no problem looking at this on their computer at home. Let's see how the next one looks. Oh, that's nice. Okay, so the way to think about it is that you've got your subunits, okay? Our subunits are green squares, and our green squares are all T-bound. And then our circles, our light green circles are D-bound. And what happens is you're going to have your subunits mostly added to the plus end. And then over time, you're going to have hydrolysis. So your D-bound will become T-bound. And then on your minus end, you're more likely to be leaving you if you're a subunit. On your minus end, you're more D-bound form, and you're more likely to disassemble from the minus end. And so this is your GTP cap. It's an accumulation of GTP-bound or D-bound subunits of the plus end. And that stabilizes your microtubule in this case, because I'm talking about GTP. So treadmilling is this concept of, oh, and I'm going to really try hard to play that treadmilling video, so get ready. Treadmilling is what happens when you've got growth at your plus end, and that's happening more rapidly than leaving at your minus end, so it gives that impression that the whole entire polymer is moving towards the plus end.
Is that only for actin?
So treadmilling, the question I just got is treadmilling only for actin? The concept when in a test tube is for both of them, but you mostly see it happening in the cell with actin because your microtubules are organized at the centrosome with the gamma tubulin ring complex. So that minus end is busy, right? You're not going to be leaving from there. You've got a nucleating protein there. So if you look at this graph, this here is showing your subunit concentration, so low concentration to high concentration, and then your elongation rate. And so if you're above, so your 0 is your critical concentration. And so if you're higher than your critical concentration, you're growing, and if you're lower than your critical concentration, you're shrinking. And your plus end and your minus end have different critical concentrations. Okay? And so if you're in the range that is basically between your critical concentrations at your plus end, which are going to be low, and at your minus end that are going to be higher, if that concentration in the tube is in that range, then you have treadmilling. Yes? Yeah. So treadmilling gives the appearance that the polymer is moving because you're adding more at the plus end and you're losing from the minus end. And this is relevant when we start to talk about cell movement. What's relevant, period, but when we start to talk about cell movement, it makes more sense. So treadmilling results from a net assembly at the plus end and a net disassembly at the minus end. Here we are again. And so this I showed you guys the other day, and here what they did is they fluorescently labeled tubulin, and they're following it over time. And you can see it gives the appearance that the tubulin polymer is actually moving. But it's not moving, it's just that you're adding more at the plus end and losing from the minus end. Should I do it? Let's do it, because it's a useful video, and if it kills my computer, we'll all say a prayer. That's not right. Okay, we're on our way. I have a ceremony for my computer shortly. Computer's like, oh my god! Yeah, let's see what happens if I do this. It's gonna be such a good video, guys. It's gonna be good. It's gonna be awesome. It's gonna be the kind of movie that you guys sleep outside in the winter to get tickets for. Oh, look, it's coming. You guys could have gone, gotten coffee, came back. Okay, while we wait for this, does anyone have any questions? Look, it looks like it's a YouTube webpage. You know what would be awesome is if it stays like that. Okay. Okay, guys. It's going to happen. For real. Let's take another 10 minutes. Any questions about this before I lose my mind? Yes. Yeah, you'll see. Watch it here. Okay. Oh, that's cute, I think. Okay. Okay, guys. Look, it's here. Okay, so this is showing actin, and your D-bound is, we're going to just keep doing this. Your D-bound, we're going to stay here all day because it's so exciting. Your D-bound is yellow, and your T-bound is red. And notice you're rapidly adding to your plus end, and you're rapidly leaving from your minus, well, not rapidly, but you're leaving from your minus end, and it gives the appearance the whole thing is moving, right? T-bound is always going to be adding to your plus end. D-bound is always leaving from your minus end. That's the standard concept with cytoskeletal dynamics, and it gives you this appearance of the polymer moving. Wasn't that worth it? Huh? No, because GTP kappa is a concept more with microtubules. Yep. Because there's no GTP with actin. It's ATP. Yes? Is there a separation point between NTP and NDP? I need you to expand on your question. Guys, I know I've given you all this time to talk about things because my computer doesn't work. But we got to respect our students who are asking questions. Let's see first if I can make this come back. Okay. The DP is what happens when you add. So you're going to have, yeah. So only T can be added. Okay. And then, only T can be added, but after it's added, hydrolysis happens. So while in the polymer, you get D-bound. The D-bound doesn't like to bind to other D-bounded strongly, it's T-bound likes to bind. And so over time, D is going to leave.
So does that mean that you get phosphorylated and that's what goes back to D?
Yeah, so D will eventually become recharged to BT. Yeah. Yeah. How long is the lag phase for when it's hydrolyzed? Okay, so you're talking about, this is what allows the GTP cap to build, right? So you're asking, how long is this lag phase? I don't know exactly, but it's long. So I feel like I know where you're going. Stop me if this doesn't clarify. So it's the lag phase that allows the GTP cap to build up. But think of it this way, it has to do the concentration of monomer in solution. If there's a ton of monomer in solution, it's going to rapidly add. So you get this buildup of GTP cap because addition is faster than GTP hydrolysis. But then as your concentration in solution goes down, there's less to be added, and that allows time for that GTP hydrolysis to catch up. Make sense? Did that answer your question? Great. Okay. Any other questions? Yes.
Okay, this is kind of a dumb question, but when you're talking about T-bound, are you talking about GTP?
Yes. The reason I'm saying T-bound and D-bound is because this kind of applies for both ATP and GTP, so I can talk about actin and tubulin.
Okay. I was just
Yeah, the concepts are pretty much the same. Other questions? Okay, it's super important that you make sure you understand this stuff because going through the accessory proteins is going to be a little harder if you don't. Okay? So that was fun. We did it. We did the treadmilling video. We survived. I should give you guys a separate assignment to work on while we wait for my computer. Okay, so this is the concept of the GTP cap, right? So with microtubules, growth and shrinkage is dependent on the presence of the GTP cap. And this is involved with this concept of dynamic instability. Whether you're T-bound or G-bound is going to control whether or not you're growing or shrinking, right? So here's your microtubule, and here is your T-bound monomers that have just been added, right? So it's a bit of a GTP cap. And so as you add more, it's growing. But like over time, you're going to have hydrolysis. And eventually, there won't be enough monomer to add, and you'll get hydrolysis, and you completely lost your GTP cap, and then you get this rapid shrinkage or catastrophe, right? So when we lose our GTP cap equals catastrophe. And then, but remember that rapid shrinkage, suddenly you're going to have a ton of monomer in solution again. And so it can get recharged to T-bound, and then you can get rescue, and you'll regain your GTP cap because you'll start growing. Yeah. Hydrolysis. Yeah. Loss of GTP cap means GTP hydrolysis because it's no more it's not T-bound anymore. Yeah.
because the microtubules are growing from the centrosome, is that the one
Since microtubules are growing from the centrosome, does that mean that the ones closest to the centrosome are more stable and T-bound? No. So the concept is the same for each microtubule, is that your plus end is where your GTP cap is going to be, and everything below it is going to be D-bound. The only difference with microtubules in the cell is that because they're growing from the centrosome, you're not losing from your myosin growing. Because you're interacting with the centrosome. So the GTP cap is always going to be at the end, and it's always going to be dependent upon concentration of subunits in solution. So over time, you're going to get D-bound, but you're not going to leave from the myosin because you're busy interacting with the gamma tubulin complex. Hopefully, as we move forward, it'll make more sense, and then come back to me if you're confused. Okay.
I just want to make sure clarify what catastrophe or what happens during catastrophe.
You just get rapid shrinkage due to disassembly of your D-bound subunits. You get, yeah, so over time you'll get growth because more T will come back and bind, and then you'll regain your GTP cap. Okay, yes?
What do you mean accidental loss?
Accidental loss, well, that's just what it says in the textbook, but my understanding of it is you don't, that it's not intentional. I don't know. It's interesting wording, right? What does accidental loss mean? I would just think of it as loss of your GTP cap. Accidental must mean there weren't enough subunits and by the fact that there's not enough T-bound subunits in solution, there's no more to add, so you're going to accidentally lose your ATP. Other questions? I saw someone. This topic always generates a lot of questions. Okay. Okay, so another way to think about dynamic instability. The GTP cap keeps microtubules growing. When the cap is lost through GTP hydrolysis, depolymerization is likely to occur. Depolymerization, shrinkage. Okay? So this treadmilling and dynamic instability, these concepts are what's important for the fact that the cytoskeleton always has to rearrange in order to respond to the needs of the cell. If you didn't have dynamic instability and treadmilling, we wouldn't be able to move, we wouldn't be able to rearrange our microtubules into mitotic spindle. We wouldn't be able to do any of that. Yes? Depolymerization is likely to occur due to loss of the GTP cap. Under what conditions would not occur? Accessory proteins are going to... So what I'm telling you about now is all the absence of accessory proteins and just the spontaneous assembly and disassembly of these subunits into polymers based on critical concentration. But when we start talking about these accessory proteins, we'll understand how actually none of that applies to the cell. Okay, it all applies. It's just altered. Okay, so actin and microtubules, they switch between growth and shrinkage every few seconds for actin, every few minutes for tubule. So this is this rapid changing, this rapid dynamic, this truly dynamic instability. The cell must use a lot of NTP to keep a constant concentration of filaments, right? Because as the D-bound subunits leave, it has to kind of get recharged to T-bound. This seems like a waste of energy, doesn't it? So much NTP being used. So what's the advantage of doing this? Why is the cell wasting all this NTP, recharging these subunits with NTP? Yep. So having it recycle makes sense rather than just creating new subunits or something like that. Yep. Okay, so go a little bit more deep. Think about what this dynamic instability does for just the function of the cell. Yep.
Would we be allowed to didn't recycle or having would be allowed to for
So you're thinking of it in the sense of you need to break it down because otherwise you'd have a lot of microtubules. Well, yes, but think about that flexibility and reorganization and how it affects the cell in response to whatever the cell needs to do. Think about that macrophage neutrophil that was chasing the bacteria. You need to be able to move around, right? You need to be able to break down actin, send your subunits to the other side of the cell, polymerize actin in the direction that the cell needs to move. You need to have this constant dynamic reorganization. Anyone want to contribute to that thought? Okay, as long as y'all believe me. Don't ever believe the professor. I just stand up and tell you. I'm just like, blah, blah, blah. Okay. I don't know. You want me to summarize? So basically, you just want to think about why is the cell contributing so much energy to this whole process. It seems wasteful, right? GTP, GDP, like constant, like GTP hydrolysis, make more GTP, you know what I'm saying? That seems like a waste. But when you consider the fact that it's essential for the cell survival to be able to respond by polymerizing actin in certain places to do certain things, for movement, for example, or to disassemble microtubules between interphase and prophase, right? You want to disassemble and then reassemble and make your mitochondrial spindle. All those things are all very important. Do you believe me? Who believes me that cell division is important? Okay. Who thinks it's totally not important? Oh. Okay, good. Good? Okay, so this constant turnover provides flexibility in reorganization of the cytoskeleton to meet the needs of the cell. These subunits, they can diffuse through the cell rapidly. And then there are all these accessory proteins that I keep on teasing you about. They're coming. Accessory proteins. All these accessory proteins that function in controlling nucleation, stabilization of filaments, they all have a huge effect on the control a cell has over its structure and function. Do you guys believe me? Sure. And so this figure is just showing you microtubules and the dynamic instability, and if you follow each one over time, you've got growth, shrinkage, this constant change. Okay, so another thing to consider, I keep saying that the cytoskeleton needs to respond to the environment, needs to reorganize based on cell cycle, but what about cell type? Right? How might actin and microtubule activity be controlled differently in different types of cells? Consider a neuron versus a macrophage. Yep.
Well, neuron, you only really need the microtubules to divide, as opposed to macrophages also to get around.
Okay, so for microtubules, neurons only need microtubules to divide. Well, do neurons divide very often?
Not really.
Not so much. So that's one thing. But what else are microtubules used for? Yep.
Stability of the cell, the neuron cytoskeleton of the cell, so the neuron like an axon needs to be stable because that's such a distance to travel, it needs to stay constant, stable, whereas an macrophage has to do things quickly, use a team quickly.
Right, so neurons, they have this long axon. And are things going to be moving up and down that axon? Yes. And so it needs that long axon, so the microtubules are going to be very different in a neuron versus macrophages, right? And then if you think about actin, which is more movement, I know you were thinking actin there when you were talking about movement. Yeah, macrophages are going to move a bit more than neurons, right? Who agrees with me that macrophages move more than neurons? Good. Right. And so then you have to think to yourself, okay, different cell types need to use their cytoskeletons differently. Well, what controls that? What regulates that? Accessory proteins. Yeah. Accessory proteins. Presence of different accessory proteins, regulation of those proteins. Yeah. Okay. Good. Those are all true. Okay. So an interesting thing is that actin and tubulin are highly conserved. What does conserved mean? Raise your hand. Yeah. There isn't much change, but go a little bit deeper. There isn't much change between species, among organisms, so they're fairly similar, exactly. And also in different cells, in different parts of cells, in different tissues, there are different, it's not just one actin. There's a lot of different, there's a whole family of proteins that also highly conserved in sequence and structure. Good. And so what are the applications of this? Something is highly conserved. It's very important. Now you can study in one organism and apply what you learn to other organisms. Yes. If it's changed, it loses a function. Yep, so let's go a little further than that. Now we're going to start to talk about accessory proteins. And so this map, whatever it's called, here is showing all, it's like an interactome. It's showing all the different proteins that interact with actin in yeast. That's a lot of proteins. Each little circle is a protein that interacts with actin. So you can imagine if actin is changed in some way, it would really affect interaction with a lot of different proteins that are needed to regulate it, right? Interesting. Okay, so intermediate filaments. So actin and tubulin are common to all eukaryotic cells. Intermediate filaments are only present in vertebrates, nematodes, and mollusks. They're not required in all cell types, only in cells subject to mechanical stress in squishy animals. So it helps to keep cells, their strength and their shape and interaction with other cells and tissues. Units that are made of monomers that dimerize and associate with other dimers to form a staggered tetramer. Notice no NTP binding, no polarity. Would you think that the concept of dynamic instability applies to intermediate filaments? No. Why?
Because there's that takes place for the GTP switching.
Right, so there's no NTP binding, so you're not going to have that GTP, GDP hydrolysis and effect on interactions. Right, and then when you start to think about the function of intermediate filaments, it kind of makes sense. But you don't want them disassembling all the time. All right. Questions? Form a rope-like filament. So it's easy to bend but difficult to break. So it provides that strength. So just like there are many kinds of actin and tubulin, should I say something really positive about NTP right now, or should I not even say a thing? Okay, I want to say something. Just like there are many kinds of actin and tubulin that localize to different cells or cellular locations, there are also different types of intermediate filaments with different functions. And so here's a list. There are intermediate filaments that are nuclear. There are ones that are only epithelial, ones that are only involved in neurons. Just to kind of keep in mind. So they provide this mechanical stability. Keratin, an example. So this is showing the staining of keratin in blue. So there are 20 different types of keratin proteins. They're cell type specific, so that is very useful. Oh yeah, see, I thought something went weird. So these keratins are cell type specific, and so they're useful clinically in diagnosis of different cancer. Keratins provide mechanical strength to epithelial tissues, right? So they anchor intermediate filaments at the sites of cell-to-cell contact of these desmosomes, and at the site of cell matrix contact, hemidesmosomes, which is down here. And if you have a defect in your keratin, it's not good, and so you can get some diseases that are associated with that. So mutations in keratin are responsible for a few diseases, one of which here out of the textbook is called epidermolysis bullosa simplex. That sounds like a lot. And it's a blistering skin disease because it has to do with the keratin's in the epithelial cells. And when it's defective, you get this blistering. ALS and Lou Gehrig's disease are associated with audit. It's like the gods of PowerPoint know that I was feeling good about things. ALS and Lou Gehrig's disease is associated with accumulation and abnormal assembly of neurofilaments. And this might interfere with normal external transport. The other thing to think about are toxins, right? Everyone loves to talk about toxins. Actin, tubulin are so some examples. Latrunculin, which comes from sea sponge, it binds actin monomers and prevents assembly. So what's the result of that? What happens when a toxin binds to actin monomers and prevents assembly? What do you think that effect is going to be on the cell? The cell can't move. Basically, anything that's going to require actin polymerization is not going to work, right? Because you need to assemble actin polymers. So one thing is it won't move. Phalloidin, which comes from fungus, binds to and stabilizes actin filaments. What do you think is going to happen there? If it binds to and stabilizes actin filaments. So you couldn't disassemble, right? You'd be stuck as an actin polymer. You wouldn't get that disassembly. You wouldn't have that dynamic instability that's required for cells to be able to respond to the environment. Coincidentally, phalloidin also has been used in biotechnology as a way to stain actin. People have conjugated fluorescent dyes to it, so you can add phalloidin to your cells. It could be, and now mind you, you're only going to do this with immunofluorescence, so these are cells that have been fixed, they're ready to be stained, they're not going to live any longer. And so if you add it to your cells, you get like red tagged phalloidin, you can stain all your actin. And so sometimes, maybe in the paper today, is it in the paper today? Phalloidin? Who read it? This is a paper that's near and dear to me, so all of you haven't read it, I'm in trouble. So yeah, so that would be bad. So then there's colchicine, which comes from the autumn crocus, and it binds to and stabilizes free tubulin. What do you think the effect is going to be there? Cancer? What did I hear? Sometimes I feel like people just think cancer. It cannot assemble. What's going to be? It can be a deeper effect when it cannot assemble. Prevent movement. Well, it's tubulin. So it's going to bind to and stabilize free tubulin. What's tubulin involved with? Cell division. So that's one thing. So it's going to prevent cell division, right? And the other thing is chromosome segregation. The other thing is going to be movement of vesicles and organelles in the cell. The trafficking of all these things that we've been learning about. That won't work. So these are toxins. So that's why they're bad. Now you know. Taxol is from the bark of the Pacific yew tree, and it binds and stabilizes microtubules. And it's used to treat breast and lung cancer. So some of these things actually have been identified by drug companies and are useful for us because specifically this one is going to preferentially kill dividing cells. Which cells divide more rapidly? Cancer cells or regular old happy cells? They divide more aggressively, and so they're going to be targeted preferentially by anti-cancer. So this is this picture showing HeLa cells at metaphase. Right, so here's your microtubules. The blue is diffuse-stained chromosomes. And what you notice is you have a few chromosomes down here, over here, that fail to attach to mitotic spindles appropriately. And so these are cells that were treated with Taxol. And so you don't have appropriate attachment to the microtubules. You also are, if you are stabilizing your microtubules, you're not going to be able to segregate chromosomes either. And so you end up with failure of cytokinesis, which is great if you're a cancer cell. Well, great for us if we have cancer cells. Cancer cells are not going to be too happy about it. Yes? How do you know that it doesn't affect normal cells? It does. That's why people get so sick when they're having chemo. Yeah, but it preferentially targets your faster dividing cells. Most cancer drugs that are more like a poison versus a very specific, like there are cancer drugs that specifically target specific genes that are mutated only in cancer cells, and that's kind of different. They're better drugs in a way. But some of these drugs that are more non-specific are going to be more toxic. Questions? Okay, so this is just some fun facts about cytoskeletal homologs in bacteria. So they have a tubulin homolog called FTSD. Its structure is similar to alpha and beta tubulin. It also binds GTP. GTP, his hydrolysis is triggered by polymerization, like us. It's orthodynamic. Its half-life is about 30 seconds. What's different is in bacteria, FTSD forms the Z-ring at the septum during cell division. So this would be like, so during cytokinesis, if we were in mammalian cells and you have cytokinesis, you also have a contractile ring that forms, but it's made of something else, right? It's made of actin. So this is a tubulin homolog. So there's a different function here. But it's similar in the fact that it's a skeletal protein, that's a homolog tubulin that's involved in cell division of bacteria. It rearranges rapidly during the bacterial cell cycle. It's involved in the pinching of the cell into two alpha chromosome segregation. So it's similar to that contractile ring, it's just not actin. Dividing chloroplasts from red algae use a protein ring that's made from the same tubulin homolog. So you can see here it organizes around the center and it tightens and pinches off into two different cells. So then there are also bacterial homologs of actin, MREB and MBEL, and they play a role in cell shape and chromosome segregation. So these here are wild type MBL. So you can notice that pretty standard rod shape. Here this one looks like it's about to divide. And here's MBL negative. Would you say there's something different here? There's a few things going on here. They seem to be stuck head to tail, end to end. They seem to be stuck end to end, and then they seem to have these kind of crescent shapes, right? So that's how people study these things. Delete it from a strain of bacteria, see what they look like. So it's involved in cell shape and chromosome segregation. There's another bacterial actin homolog CAR-M, which is involved in plasmid segregation. So in it also, there's monomers, and then there's filaments. That same concept of polymerization or filaments. And what this CAR-M does is it's going to interact with the origin of replication on these plasmids and actually push them apart prior to cell division, so that when cells do divide, each daughter cell has the plasmid. So interestingly, CAR-M is included by the plasmid itself, so it allows itself to be distributed. Questions? So then there's another bacteria, Caulobacter crescentus, and it expresses an intermediate filament homolog called crescentin, and it's responsible for its crescent-like shape. And so here you can see in the pink, I guess, or red, that's standing for crescentin. And so when you have crescentin, you've got this crescent-like shape, and when you delete crescentin from this bacteria, it would straighten out. So you might be like, so what does this have to do with anything? Well, it's beta conserved more than one would think. And it's just interesting to see that bacteria, which are quite far from us, have similar proteins that play a similar role. Shape, vision, plasmid segregation. Great, so what's the relevance of these homologs in bacteria? So just think when you're studying, you're thinking about this, think about it. How are their functions similar to eukaryotic homologs? How are they different? What functions are present? What functions are missing, right? So we saw functions in shape, in plasmid segregation, in cell division. What function that we know microtubules and actin play a role in that we didn't see here with bacteria? What functions are missing here? Anything? Or is it all exactly the same? Yes. Movement? Here we didn't see movement. I was thinking of something else though. Transport, vesicular transport is definitely missing. We have organelle transport, right? Missing. Why? We have organelles, right? Okay. So the cell, so a lot of what we've learned about so far is about how these different monomers behave in a test tube without accessory proteins, these famous accessory proteins that I keep talking about. But what happens in the cell needs to actually regulate the length of its skeletal filaments and its accessory proteins that do this. They either bind to filaments or free subunits and control their function in some way. They modify dynamics and structure, and so it's important to understand these. So I'm going to direct you guys to panel 16.3 in the textbook. I do not believe it is available on the online version, so please come see me if you cannot access it in some way. It's useful though. And there's a really nice illustration of all the different accessory proteins and their functions and how they interact with monomers or subunits and polymers. It's always nice to have everything in one place. Okay, so we're going to start with talking about proteins that nucleate. What is nucleation? Who remembers? That was like eons ago that we said that. Yeah. Great, so it's the formation of a cluster of subunits that can be easily added onto. So it's a stable number of subunits that allow for growth of the polymer, right? So how does nucleation regulate microtubules in action? What happens when you nucleate? You drop. Okay, you can get polymerization after nucleation. So we're going to talk about proteins that are nucleating. Okay, so the first example is gamma tubulin, and it's involved in tubulin. It's involved in nucleation of microtubules as part of the gamma tubulin ring complex, or gamma TERP. So you might see on the exam, I might just write gamma TERP. Just know that's gamma tubulin ring complex. And so what happens is you've got your gamma tubulin, and it forms this stable nucleus, upon which microtubule subunits can assemble. So you can imagine if you add a lot of gamma tubulin to your test tube full of tubulin monomers, what's going to happen? Will the tubulin monomers stay tubulin monomers? You'll get nucleation because you've got gamma tubulin, and then you'll get growth of microtubule. So the centrosome, it contains more than 50 gamma tubulin ring complexes. So here's your centrosome, and there's a ton of these gamma tubulin ring complexes. So that's why whenever you're looking at a cell that's staining for tubulin during interphase, you're going to see microtubules radiating out from an area just adjacent to the nucleus. And so that's your centrosome, and it's kind of like the birthplace. Right? Your microtubules are, and so they're going to organize here, and they're going to grow, pushing outwards against the cell membrane. And that pushing outwards on all sides is going to center the centrosome. These centrosomes are going to duplicate during interphase and prophase, and move to, did I just see? Is it flying by? I'm not using my line. And they're going to move to the opposite side of the cell, opposite side to the nucleus, and form the mitotic cell. Okay? Yeah, so that was wrong. Okay. So how are we feeling? Everyone having fun? How do you guys feel about the introduction to your first accessory protein? It's like a gateway accessory protein. Nothing. Nothing. I got like one chuckle in the front. Okay. Okay, let's stop here. I'll see you guys later.